Okay, we are back and we are gonna go back. We're gonna really go back to the basics of cell biology. So we got, let's see here. I don't know if you remember this, but we're made out of these things called cells. Okay, and cells got a membrane. Oh, whoopsies. Right? They got this little cell membrane. A cell membrane. And deep inside, we got a nucleus. All right? And inside of this nucleus, tucked all up in here, what do we got? We got a bunch of nucleic acid, also known as our DNA, which contains things like genes, which do things like code for protein. If you guys remember this, DNA usually codes for RNA, and then that codes for protein. Okay, that's called the central dogma of biology or molecular biology. Okay, so we got a nucleic acid that makes a protein, all right? That's what your genes are. Your gene is a part of your DNA that codes for something. Codes for an RNA, which then is converted into a protein, right? And if we zoom way out and go way back over here, all this will start to make sense again. We had things like, we're going to come back to this idea. We had our DNA wrapped around this stuff. If it's tightly wrapped, the gene is off. If it's loosely wrapped, the genes are on. Things like your thoughts can change that. Can change whether your genes are off or on. For example, if you're really stressed out, you're going to have all your stress genes on. Whereas if you're real relaxed and chill, you're going to have your chill genes on, your positive genes. Okay? This DNA codes for RNA, messenger RNA, which can then leave the nucleus, and we make something called a protein either in the cytoplasm or over in the endoplasmic reticulum, right? And then this protein is in a vesicle, goes over to the Golgi, picked up by another protein. The vesicle's picked up by another protein called a kinesin motor, right? And then it's dumped down at the edge of the cell. All right, that's our kind of world we're living in again at the cellular and molecular level. All right. Went through all these organ systems. And now we is back to life. Back to reality. Okay, so we're going to be talking about DNA. And DNA is, like I said, it's in these couple different states in your body. And I just want to kind of give you some mind-blowing facts here about DNA. So each one of your cells is like approximately like 20, they vary in size, but they can be, some of your cells can be a meter long, but 20 micrometers long. They're small, very small. Micro means like 1,000 times. There's 1,000 micrometers in a millimeter. You can see a millimeter with your eye. You can't see a micrometer with your eye. The lowest you can see is about 50 micrometers with your eye, something like that. Okay? It's very small. Very small. But each one of your cells holds about 2 meters of DNA. Like if you were to take the DNA and lay it all out end to end, all your chromosomes, it'd be about 2 meters long. Okay? You are made out of about 37 trillion cells. Cannot type. 37 trillion cells. Not all of them have DNA. Like your red blood cells don't have DNA, and most of your cells are actually red blood cells. But then your muscle cells have multiple nuclei, and some other cells have multiple nuclei. So I'm just going to count them all as having DNA. We're just going to cheat. You have 37 trillion cells, which means that you just take this number, multiply it by 2. You got 74. I'm kind of rounding up, or would that be 76? No, 74 trillion meters of DNA. Okay? We're going to have a little fun here now. The sun is about 150 billion meters away. All right, it's pretty far. It's not that far, actually, though, in the grand scheme of things. Space distance. Which means that your DNA could shoot to the sun and back 250 times. Or it could shoot to it, boom, 1, 2, 500 times, just going to the sun, the distance of the sun. 500 times. Call that one-way trips. So it makes sense. That's crazy. That's far. It's 92 million miles away, roughly. And it could do that 500 times. That's insane. That's insane. Okay, even more insane. Pluto's even further. Your DNA could shoot to Pluto 14 times. One-way trips. So, that counts as 1. Counts as 2. You could have 14 sections of your DNA reaching Pluto. I don't know exactly how far away Pluto is. I don't remember. But I did that math a while ago, and it's pretty crazy. Okay, even more crazy. Light travels at a speed. It's very, very fast. It's like 186,000 miles a second. It's really quick. Goes around the Earth like 7 times, in a second. Yeah, really fast. It's really fast. It would take light. It would take light 2.7 days to travel the distance of your DNA. That's crazy. If you ever think that you're not incredible, you are. You are incredible. Just kidding, you're not incredible. Blah, 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 blah. Okay, let me pull up a little picture here. That's just some fun, interesting facts about DNA that I find mind-googling, boggling, bonkers. Okay, we got this little picture here, right? And I've showed you guys this before. This would be a nucleus and all the DNA's in there. And if we crack that boy open, when it's all condensed, it looks like this. And each one of these things is a chromosome, which is made out of strands of DNA. We have 23 chromosomes. We have 22 somatic chromosomes and 1 sex chromosome. Or 2 in the case of males. This is a female, 2 X's. Okay, so we're diploid. We have 2 versions of 1, 2 versions of 2. Well, this is very tightly packed up. This is how DNA is in the nucleus. It's very kind of spread out. You can't see the individual chromosomes because some of the genes are out. It's open. It's ready to be read. It's ready to be turned into RNA so that can be turned into protein. But when the cell divides, it packs it all up super tight like this. So we're going to talk about this little process. How does this work? and like what really is this thing that we're looking at. So let's go back to the beginning of just like, we're just going to look at some DNA strands. Okay, so we got a DNA strand, which is a double helix, like so. And in the middle of it, we've got bases. I'm not going to draw them in different colors, like A binds to T, and T binds to A, G binds to C, and C binds to G. So we got a single DNA strand. So if you were to unravel all your DNA into this form, into just this strand, and stretch it all out, it would take 2.7 days for light to travel the distance of your DNA. Like, that's the code that makes all your cells and gives you all those instructions for like what your kind of traits are going to be. Okay? It's kind of bananas. Okay, now we're going to zoom out a little bit. We're just kind of like real zoomed in on a little section. We're going to zoom out, and what we notice, we've already talked about this before, but there's also protein that the DNA is wrapped around. I'm going to actually just copy this. Like so. Okay? So the DNA actually wraps around this protein. It actually wraps around twice. I'm just going to do it once. But it actually wraps around twice. Okay? So this section right here is like looking right there. And then if we zoom back, we can see that there's also these things called histone proteins. Histone proteins. And this would be DNA. All right. DNA wraps around histones twice. Okay, and this is called chromatin. Specifically, this is called chromatin. Specifically, it is called euchromatin. Euchromatin, meaning true chromatin, because the DNA can still be read. Genes are on in this loosely packed DNA state. So the machinery, the polymerases that can come in here, and go and unravel that and turn this gene into RNA because it's nice and loosely packed. Okay? All right. Now we can take this DNA and we can further pack it. So further packing. And it'll look something like this. I'm going to grab one of these bad boys. Boom. And spread those out just a little bit. Okay. And oops. And bring these down to the bottom. Beautiful. Then DNA is still wrapped around this. And again, it's wrapped around twice around each one of these, but just for simplicity, I'm doing one. like so. All right, boom. Okay, this is again DNA and still histones. Histone. This is further packed, right? And it's heterochromatin. Heterochromatin. It's not true chromatin because the gene is off. We already talked about that, but it's been a while, and you can start to really appreciate how packed up this is. We've gone from, you know, a very linear piece of DNA like this to, okay, we packed it up a little bit, and now we've really started to pack it up. Okay, let's keep packing it up and packing it in. If you try to battle me, that's a sin, bro. Okay, and further pack. So now I'm going to show you a DNA strand. Do it like this, I guess, because it's that DNA around those proteins. So this heterochromatin is what I'm showing you. Will then loop around. Make sure it's kind of all connected. So we're just further packing it by creating these loops of heterochromatin, of this. These loops, that's made out of this over here. Okay, so this is loops of heterochromatin. Okay. And we continue this kind of idea even further. Continue to pack and continue to pack, and we also zoom out. And we get something that looks like this. So these are those loops. And it's like even more looped than this. Obviously it's three-dimensional. I'm not drawing a three-dimensional kind of thing here. Boop, boop, boop. Okay, beautiful, beautiful, beautiful. Loop, 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 loop. Okay, very good right there. And it gets kind of thin usually at a point, and then it'll get thicker. Boop, 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 loopy, 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 loopy. Oh, that was a fiasco. It was a loopy fiasco. Kick, push. Kick, push. Very relaxing, actually. All right, okay. Okay, boom. What do we got here? Oh, that looks kind of similar to this thing, doesn't it? This is one of those right there. So this is a chromosome. All right, so this is a chromosome. And this stuff is really confusing. This is a chromosome. All right. And what we're going to do before we divide a cell is we're going to make a copy of it. Okay? I'm going to give you a little anatomy here of some DNA. So this is a chromosome, right? And we would have two versions of this. We'd have this version, and we'd have another version of it. Go ahead and bring this boy over here. So this would be version 1. which contains whatever alleles it has. And this would be version 2. Version 2. Okay, so that would be like right here, right exactly what we're looking at. And you see these also have this kind of like little midpoint right here. Right there. That's called the centromere. All right. So what happens is, I've not really talked about the cell cycles yet, but before a cell divides, it makes a copy of all of its DNA. Boom. And this is a copy now. And right here, we got a specialized little structure. Okay. I'm going to copy this bad boy. Oops. No. I do not want to do that. Okay. Boom. DNA has been copied. Oops, that doesn't look like I want it to. Okay. Okay, there's a lot of definitions here. Okay, so this is a chromosome. One of these things is also called a chromatid. Chromosome or chromatid. All right, and these two together these are together. They're sharing this centromere. These are sister chromatids. And we'll review all this stuff, so don't worry. These are sister chromatids. Okay, this is all important for kind of understanding the differences between mitosis and meiosis. This is a centromere, not to be confused with the centrosome. And each one of these, since they're both the same, they're both different versions of the same chromosome, these are called homologous pairs. Homologous pairs. Homologous, G-O-U-S. Homologous pairs. Okay, and actually, I want to swap this just so this is a little more clear. I'll put this. I'm going to put this up here. Homologous pairs, I'm going to put it up at the top. Okay, so those are sister chromatids. One of those is a chromatid. We got version 1 here. And we got version 2 of the same chromosome, and these are homologous pairs. Okay. All right, so let's kind of do a little review of all this stuff. All right, I got another little picture here I want to pull up. Whoops. Like so. This is everything that we just kind of went through. So this here is a DNA strand, right? DNA strand. And then DNA wraps around these histones. So you can see this wraps around these histones, and you see it wraps around 1, 2, 1, 2. So this would be chromatin or heterochromatin, chromatin right there, which is DNA around protein. That's chromatin. And then we have our loops of heterochromatin. So it's, you can see it actually spirals around like this. And then it forms these loops. And then we got this thing here. And this thing is a chromosome. Or or a chromatid. Okay? And if we were to do this, and draw another one of these in here, like so, these would now be sister chromatids. Okay? And this thing here is a centromere. Okay. All right, let's define some of these terms, and then we'll get into the next lecture. All right, so I'll kind of keep, actually I'm going to move this over, and we'll keep this up so we can see it. Maybe we'll have them both there. That's fine. Okay, so a chromosome. Chromosome is a complete set of DNA that has a specific size and contains specific genes. Kind of a weird definition, but that's what we're going to go with. I'm going to actually kind of do a sticky note. I think I can, but it's okay. We'll just do that. Okay, so this is a chromosome, contains specific genes. Always, like, let's say like our hemoglobin gene is on chromosome 1. I don't know if it's there or not, but it's always going to be on chromosome 1. Okay? All right. A chromatid, this is the same thing as a chromosome. However, it is the single half of a duplicated chromosome. That makes sense. Also kind of weird definition, right? This whole thing is not a chromatid. Only this is a chromatid. And these two chromatids are sister chromatids. This one and this one would be non-sister chromatids. We could put that on there too. This one and this one are non-sister chromatids. They contain different versions of the same traits. Okay? Okay, so sister chromatids. Sister chromatids are copies of the same chromatid, and they are connected by a centromere. Mouthful, lots of terms. You're insane. Me too. Biology. I just want to kill my veins. Okay. I'll do non-sister chromatids too. Non-sister chromatids are chromatids of the same, of different versions of the same chromosome. This is, I feel like I'm just going to be confusing everybody, but that's okay. Don't worry about it. Okay, that's a non-sister chromatid. Okay, homologous pairs. Homologous pairs. Homologous pairs are two versions of the same chromosome. Duplicated. Duplicated versions of the same chromosome. So it's the two versions. Another way to think of it is two versions of the same chromosome. So we have version 1 and version 2 because we're diploid. Those are homologous pairs. Okay. Centromere. Centromere connects sister chromatids together. Okay, that is this thing connects these sister chromatids together. Okay. We have not talked about this, but a centrosome organizes the microtubules to form the spindle. Microtubules come out of this to form the spindle. Don't confuse centromere and centrosome. And the centrosome is made out of 2 centrioles. I know. Okay, and last but not least, this one's easy. Chromatin. Chromatin. This is DNA wrapped around protein. Specifically, histones. Okay, so that's chromatin. Okay, that's a lot. That's a lot of terms. Make sure you know the differences between all those. Especially like chromosome, chromatid, sister chromatid, homologous pairs, centromere, centrosome. Also chromatin. You got to kind of know all those. You really do. Non-sister chromatids, that's easy if you know what a sister chromatid is. Okay? Let me know if you have questions about that stuff. Okay, now let's put all this kind of together and start talking about this process here in the next one about how do we go from this single stem cell to making a human? This process of mitosis. What are we actually doing here? Okay, I'll see you there.